I'm here. We can get started. So.
Yeah.
Do we have people who's on? Bunch of people.
Yeah. Get them out.
Right. So, welcome to the Southern California Environmental Health Sciences Center seminar. This is the last the last seminar of the academic year. And it's my pleasure to introduce Dave Conti. Dave and I have in common that we both were born in Denver and grew up in Colorado. We went to the same university at very different times, and we both ended up here at USC. Dave's, as most of you know, Dave's a biostatistician. He's doing really innovative work in a number of areas, methods work with application to environmental health and other issues, and he'll be talking today about some innovative work on polygenic risk scores and their how to use environment in the context of polygenic risk scores. So Dave,
Thank you
for coming. Take it away.
Okay, thank you very much. So I'm going to talk about sort of the interface of the polygenic risk scores, non-genetic factors, and populations. So this talk will be less statistical models and more just interpretation and use of models like that. And in some ways, it'll also be a primer on PRS and applications to things. And a lot of this has to do with my role as part of the Primed Consortium. 1 second here, everybody. There we go. Which is an NHGRI funded consortium for developing polygenic risk scores across populations. And this is really the goal of this is how we can have risk prediction models or applications of polygenic risk scores work for all individuals. And so that includes gathering the diverse data sets, developing new methods, collaborating, and then ultimate goal of improving health and having these polygenic risk scores impact how we do clinical and screening protocols. As far as this consortium, I lead the Prime Cancer part of this, the PI of that, and it's leveraging diversity in cancer epidemiology cohorts and novel methods to improve polygenic risk scores. And it's 3 simple aims, really. And the first one is to construct the polygenic risk scores. Second is to how to evaluate these across populations. And the third is really how to estimate absolute and excess relative risks with more joint models. And that's really what I'm going to focus on today, or get to that point, and talk about some of the issues in thinking about how to create these joint models and apply them. So with that in mind, with Prime Cancer, we are leveraging about 6 or 7 different prospective cohorts in cancer. So we have the multi-ethnic cohort, which I'll talk about today. But we also have access and collaborators from the Kaiser Genetic Epi Research cohort, JERA, the 3 different Harvard cohorts, the Women's Health Initiative. There's the Women's Health Initiative. Then there's the two Harvard cohorts, Nurses Health Studies and the Health Professionals Follow-Up. And then we also have the PLCO trials from NCI. And that includes about 300,000 individuals in total, 100,000 non-European ancestry individuals with 91,000 incident cases. And the idea of all of this is these are all true cohorts, so we can do prospective analysis and estimate absolute risk from that. And in addition to that, we have access to the individual level data that comes from cancer site-specific consortiums. So NCI has sort of over the years created cancer site-specific consortiums that have gathered all the world's GWAS data for each different site. And there's a number of us here at USC that are involved in several of these different consortiums. And I'll talk specifically about the prostate cancer one today and how we've used that data. So just, in short, our goal is to do risk prediction for all individuals. And in doing that, we have to consider sort of the genetic variation of individuals across populations, or also add mixed individuals. But we have to put this in the context of non-genetic factors, cultural factors, race and ethnicity, different nations, other things and other factors that we need to consider in creating these risk models. And so when we look at the process of developing PRS for screening, there's several different steps, and I don't want you at this stage to really focus on the details of the slide because it's small in the room here. But the first step is for discovery. How do we discover the SNPs that might be involved in a polygenic risk score for genetics? And we have these consortiums, and they have GWAS data. They share summary statistics. You can get this; they're publicly available, and there's statistical methods for doing this type of analysis. But the thing to think about for this step and throughout this whole process is, how do we define populations, or how is the data aggregated by population? So most of us, in terms of GWAS studies, they're really sort of aggregated on continental ancestry by design in terms of early days of GWAS, and I'll talk a little bit about that. And then as we start to develop a polygenic risk score, we want to evaluate it. And the first step is to evaluate it at the population level, really things, statistical metrics such as R squared or AUC. How well does it perform in terms of risk prediction or overall in a population? And then we get more into the individual level evaluation, which is more in the realm of sort of absolute risk, or how we would predict for an individual and a model that would do that. And that's where I'll get to sort of at the end of this talk today. And then the ultimate goal would be to sort of have a prospective screening trial where we take the polygenic risk score into the population and evaluate how it can impact screening, treatment, and other paradigms from the standing. But again, at each one of these steps, what's the population, who are the individuals, and how do we apply these models? So from a genetics point of view, we can sort of break the world down into two components. There's the genetics, and then there's everything else, okay? And I'm going to use a DAG here to describe a lot of what I'm talking about, but it just, don't focus too much on the details, but it's really using this DAG to describe the relationships of the different components in a polygenic risk score, but also the components as we expand out into non-genetic factors to think about how we do this. And so, really we have this genetic component, and it's both sort of here in a circle and in a square, because we have some of these causal factors that we've measured, and there's others that we don't know, right? And then everything else in terms of the non-genetic factors is this X, right? And from a statistical genetics point of view, those are the models that we use. We really just look up and we sum up all the genetic factors, and we leave sort of a random effect for the environment, and we go on our way, right? So there's a little more nuanced as we go forward with this, and we wrote about this DAG several years ago in the American Journal of Epi, of how sort of we don't measure all the causal variants. We measure 1 million SNPs along the genome, and those SNPs are correlated with the known genetic factors, right? And they're correlated through evolutionary history, and we call this linkage disequilibrium, the amount of correlation that they are in a population. And it's this ability, this correlation, this line here, that allows us to do GWAS studies, right? So we don't have to measure the causal variant. We can just go through and just use these as signposts along the genome, test all of those, and we get these wonderful Manhattan plots. And this is just a plot from one of our prostate cancer GWASs, and you can see that we have on the y-axis the negative log p-value, so we have lots of statistical significance, and we can pick out sort of these regions that harbor putative causal variants, right? And the reason that there's a peak there is because there's a lot of SNPs that are correlated with each other surrounding one or more causal variants, and we have to identify those, right? And that they're correlated through that evolutionary history, right? And that really gets at this idea of locally around that region, there's local ancestry that will define that correlation. Okay, and that's what's represented here by this L. And so if you think about, you have like a single population and you get two different chromosomes from individuals, and there's crossover events from your parents, and that sort of shuffles the chromosome segments that you get from your parents in terms of the chromosome segments you have, right? So if your parents come from a single population, then those segments will be representative of that population. They will have similar allele frequencies and similar arrangement of the variants along the genome. Okay, if they come from different populations, then those segments will be different in terms of the allele frequencies in those respective populations. And we can identify those segments as local ancestry segments. So just as a cartoon about how this is, there's three populations represented in these chromosomes. There's a sort of a yellow, a purple, and a green segment that comes, and you can imagine that here's an individual that's admixed in their history, in their ancestry. They've had different individuals come from these three different populations, and they've all contributed different parts, okay, in terms of these local ancestry segments. And we can identify those, but we can also just average across those chromosomes, and we can get the idea of global ancestry. Okay, how do we summarize someone's global ancestry or diversity along that? And so you can see that here in sort of looking at different chromosomal segments, an individual could be a certain percentage from the yellow population, the green population, and the purple population as we go forward. And so represented in our DAG, we have this global similarity or this genetic similarity, and that's capturing this ancestry as a whole. Now, we often don't measure it as something as averaging over the local segments, because there's some problems with that in terms of identifying what populations are ancestral. And usually we do it through a set of ancestry informative aims to estimate that kind of ancestry. And so we can do that in the population, and I'll show some examples of that here in a little bit. But we have to realize that that global similarity that we estimate is highly correlated with race and ethnicity. Okay, that both of that and the shared history of an individual is also defined by their race and ethnicity. And race and ethnicity are also associated with these other non-genetic factors that we could measure, these true risk factors. And that induces a correlation in terms of this genetic similarity and how it's related to non-genetic factors as well, okay? And that's really gonna be the crux of what I'm talking about, is how these different things are related to each other and how we can parse that out, okay? So it's this confounding triangle that sort of induced here that really led us to sort of use genetic similarity as a confounding and adjustment variable, right? So when we test a genetic marker, we always include genetic similarity as an adjustment variable. But it's also sort of this differential LD across populations and this difference in terms of allele frequencies and confounding that led GWAS studies early on to really stratify by race and ethnicity or continental ancestry. So again, that goes back to why NCI has these consortiums really designed around, you know, summary statistic GWAS of race and ethnicity. And so that was how things were done maybe 15 years ago when GWAS got started. We believe, well, we really need to have homogeneous populations to find things in genetic studies. And that had a big impact on the studies that we have today in terms of genetic GWAS. And so this is a figure showing the growth of the number of individuals included in GWAS over time. And you see that it's growing exponentially, but it's predominantly from this red group here, which is individuals of European ancestry. So we've grown GWAS, but we've grown it with individuals of European ancestry. And that proportion that it's in GWAS is certainly more than what's represented by the global population. Okay, there's been growth in non-genetic or individuals of non-European ancestry, but it's much smaller. So if we're discovering variants that are in individuals of European ancestry, and then we construct a risk prediction model from that, how well will it perform in other groups, right? And so there's a paper, again, by Martin that came out and looked at this, and relative to applying those risk prediction models, okay, and this is across 17 different traits, in relative to how well it does in Europeans, you can see when applied to individuals in other populations, it does not do as well, okay? And pay particular attention to how well it does in African populations. So we develop a risk prediction model, and it does not do well in other populations. So if we apply that, it could actually increase health disparities. Okay, why is that even more important? Well, for prostate cancer, we know from SEER age-adjusted incidence rates, prostate cancer has about a 1.5 to 2-fold increase in incidence relative to, for African Americans and African ancestry individuals relative to white individuals. So again, this would actually make it much worse if we developed a tool to risk predict and didn't work well in African individuals. So to overcome this, and really a lot of this research is driven by Chris Hyman over the years, is to increase the diversity for the prostate cancer GWASs. And this is just showing sort of at the top up to that point, this is a few years ago, where we were in terms of the number of individuals overall and how that was split across the different continental ancestry groups that we had access to. So you could see that we were about 100,000 cases, 100,000 controls split across the 4 different sort of continental ancestries, still predominantly European ancestry individuals, but we had at that time the most diversity in terms of cancer GWAS. And then we went out and when I say we, I really mean Chris, going out and adding more individuals. And so 11 new studies, a 57% increase in non-European cases, and an overall 128% effective sample size increase. Okay, so that was the GWAS data that we had access to. And now we want to construct our genetic risk score. And just to be clear about what genetic risk score and also called the polygenic risk score, usually the difference is they're really one and the same. Often genetic risk score is for when you have a small number of variants. A polygenic risk score is when you have lots of variants. When a small becomes a lot is up to the interpretation of the user. And so the genetic risk score is really just a sum of the weighted risk alleles. So we have G here, which is the number of risk alleles that we've identified for the GWAS, and then we weight it by some weight, and then we get our genetic risk score that we can use in our studies, right? So the question from a statistical point of view is what SNPs that we're going to include, and then what weight, right? And that's been the subject of lots of work, and most of that then is sort of from that one part of the DAG that I showed earlier. And it really comes down to sort of two sort of themes or trains of thought. Is the goal really to do the best job that we can to pick the putative causal variants? So we're doing some sort of statistical model where we're selecting a small number of variants and we believe that those are really causal. Okay, that might be a good frame of way to do it if you think that if we have the causal variants, it'll transport across populations better. Okay, that's been one of our thoughts. The other is to weight the entire region. Don't worry about identifying the specific ones. Let's throw them all in the model, weight them appropriately. And that's really sort of Ridge. There's a method called LD score. There's PRS, CXS. Throw it all in there, weight everything, and we'll get a polygenic risk score that way. Okay, so that's well and good on a single population, but then there's issues of how we do that across populations. And again, are there different SNPs selected or different weights? How do we then approach that across populations? And that gets into another goal of what are we trying to do with these genetic risk scores? Do we want a population-specific genetic risk score or an ancestry-agnostic genetic risk score? And so there's been work in type 2 diabetes, for example, where they've gone and they've used only individuals of African ancestry and have developed polygenic risk scores there. And they show that that works well for African ancestry populations, better than a multi-ancestry type of thing. However, that limits it to just putting people in that bin a priori. Right, so we've been more of the belief of sort of creating an ancestry agnostic one where any individual comes in regardless of their ancestry, and this risk score would work for that. So that's what we did with the latest data. We have a method that we call MJAM to do this, multi-population joint analysis, and we identified 451 prostate cancer risk variants. It uses a single set of SNPs, 451 for any individual, and it used a multi-ancestry weight to get that estimate. And so we have several papers on sort of the statistical methods behind that development. So then we have to ask ourselves, well, what does this look like? We have the SNPs, 451. We were really aimed at trying to identify which ones are the putative causal variants. So hopefully some of them are genetic like causal. Many of them will just be sort of NLD with those, but we can use that to construct the polygenic risk score. So now we have to deal with sort of how it's related to population distributions. And if we sum this up and we look at our genetic risk score, this is the distributions we get across 5 different populations here in the multi-ethnic cohort, which I'll go into in a second. And we can see that there's a shift, okay, in the distribution where there's a higher distribution, mean distribution for those individuals of African ancestry. Okay, and so we're very tempted to say, look, we're capturing something there, right? And we line that up with what I showed earlier in terms of the SEER incidence, and we think, this distribution can describe those differences. Okay, but we have to be really careful because we can't interpret the distribution of the polygenic risk score, okay? Because it's confounded by evolutionary history. It's confounded by the LD. And there's been many studies, and this is just highlighting some, and I don't need to go into the detail, but the highlights are said in these comments here is as the linkage disequilibrium between causal variant and associated varies across populations, you're going to get just a shift in that distribution that could go either way. Okay, but on average, it actually shifts African ancestry distributions up more because of their variation in their LD. It also impacts the weight estimate we get from that. And so here's examples of two PRS distributions, one in cardiovascular disease, the other in type 2 diabetes, where we do see the shift of the distribution being higher in African ancestry populations. Okay, so we can't interpret this distribution. So what we normally do is standardize that distribution by race and ethnicity or genetic similarity. So statistically, we subtract off the mean in a population and we divide through by the standard deviation, right? We can do that by race and ethnicity as that population, or we can do it by the continuous genetic similarity values that I'll talk about here. Okay, so just looking over the years of how we've increased sort of the performance and the discovery. So this is a single slide that I can't maybe underemphasize the amount of work and money that has gone into this, because this is the sort of prostate cancer GWAS over the years, starting back in sort of 2014, where we had initially about 40,000 cases in controls. We identified 100 different variants. Now, through the years, the next thing we did were population-specific GWAS to discover population-specific variants, and we had several different ones of those. And then a couple of years ago, we combined all that data using sort of the earlier versions of what I just talked about with MJAM to sort of identify 269 variants, and then the latest here for 451. So through the years, we've increased sample size, we've increased diversity, and the impact of that has been great. So on this figure here, we have the x-axis is the odds ratio per one standard deviation increase in this PRS, okay? And so that's common across all the populations. We have it separated into continental ancestry, Europeans, African, Asians, and Hispanics. And then we have the 4 different GWAS over time. And you can see within each population, as we increase sample size and discover more, we've increased the performance. We've increased the odds ratio for a 1 standard deviation increase in PRS, right? And we've done that for all populations. And proportionally, as we've increased diversity, we've increased it in the non-European individuals. And so 2 things take home here is the odds ratio, the 1 standard deviation odds ratio for prostate cancer we have now is above 2 for all populations that we've examined. So 1, 2 is high for all cancers. So it's the highest among all the cancers. And we're unique in that we have sort of a consistent estimate across populations. So diversity matters, having more people matters, having bigger sample size in those populations matters as well. So, yes.
I just have a question because
Yeah.
as it goes to different slides, so I guess I'm just a little bit just a little bit clarification on the use of Hispanic versus American, only because it's because here, right, like it's discussing sort of continental sort of ancestry, right? Which in that case, American would make sense because Hispanic usually, for some demographic reasons, would be a mixture of indigenous and European.
Absolutely, yeah, great, no. I've glossed over that and I'll get to more details on that difference, because Hispanic as a population is a population that includes admixed individuals by definition. And so there are individuals within self-identified Hispanic population, Latino populations that are 100% Amerindian or American ancestry, and individuals that are 100% European ancestry, and then African ancestry as well. When I refer to continental ancestry, it's usually just European, African, and Asian as an ancestry, as well as self-identified of where they came from in terms of their self-identified sort of location. Hispanic really doesn't fall under that. And so it's sort of a 4th different group. So yeah, it's really 4 different populations, whether it's ancestry and self-identified.
Yes, so seeing these, for example, this was,
Yeah.
it's like continent bonds except for, absolutes.
Except for, yeah, exactly. And that is an excellent point and one that we'll talk about here in just a second. So, okay, so this gives us a relative risk, right? A 1 standard deviation within a population. So put people into a band and we have sort of what's that increase in that. But what we really want to do is define absolute risk, okay, overall. And to do that, we have models like this, where we model the risk of somebody who's exposed is going to be equal to the baseline risk times this relative risk that we just talked about, okay? And we can estimate these models from reference data. And we can take that from the SEER data as an example for cancer. We use ethnic-specific incidence rates, ethnic-specific mortality rates. So those serve to give us a construction of the baseline. And then the work that we just showed, in terms of the relative risk, we can use the polygenic risk score that I just described. We can use either ancestry-specific estimates, or we can get a single multi-ancestry. So take a meta-analysis across those different populations that I just talked about, right? So it's about 2 per 1 standard deviation. And we can construct figures like this, okay? And so just to give orientation here, this is the cumulative age-specific absolute risk for an individual, going from age 50 up to age 85 there. And it's split into sort of 2 different figures here. If we look at the top one here, this yellow distribution, that's the genetic risk score distribution in individuals of African ancestry. Okay, if we define a line of a percentage where there's a 2-fold increase, that captures 16%, okay, of the population. We define it by the control, the population level. And those individuals, then, if we model that, that's this yellow curve, and they get up to about a 23% or 23% absolute risk, lifetime cumulative risk. Okay, how does that compare then to the average in those populations? Well, that's 11.6%. So these individuals at a high genetic risk reach a lifetime risk that the average person does about 19 years earlier. Okay, we can do the same thing in European populations and get that, and it's about individuals that are at a high genetic risk get a lifetime risk of the average, the individual, which would be about 8% here for Europeans, 16 years earlier. So this has implications for screening. We should start screening for these individuals earlier, right? Now, the problem with prostate cancer is we don't actually know what we would do once we identify those individuals, okay? Because we don't do, is it PSA type of things, is it others? If you think about colon cancer, there's actually, you can take a colonoscopy. And so there's definitely a sort of an implementation plan there of identifying individuals at higher risk and doing earlier screening. And this is one of the reasons sort of that you're now seeing sort of proponents for earlier screening in colon cancer. Yes.
So does anybody, the problem with PSA is it's not a great screening test.
No.
So has anybody looked at how predictive performance of PSA works in the context of different polygenic risk scores?
Not as in depth as we'd want. And there are screening trials that are looking at that, and it's complicated because you need to do the prospective screenings trial where you've actually done the right thing with the PSA screening. The real question for prostate, I'm going to go off a little bit, but the real question for prostate cancer is who has aggressive prostate cancer and dies versus who has just sort of localized prostate cancer from a screening thing. Of the 451 variants that we've identified, 50 of them approximately are also PSA variants from their GWAS as well. If we pull those variants out, our genetic risk score can identify a slight difference between aggressive and non-aggressive because it removes a screening bias from PSA. And so those individuals who are higher on the genetic risk score for PSA are more likely to be screened, more likely to have non-aggressive prostate cancer. So there is some indication that this genetic risk score works for aggressive prostate cancer, okay? But it's a little bit more nuanced than that. But to answer the question about does a genetically informed screening protocol with PSA help prostate cancer survival? That's an unknown. Yeah, and it's part of why we're doing the RESPOND study, which is the PO1 that we have of 12,000 African-American men with prostate cancer.
Hi,
Okay.
hi, David. It's
Yes.
Claire Campbell online. I had a quick question about how you defined these groups, these ancestry groups, because usually you have like a threshold of like the ancestry that you use for them and how you group them. I'm wondering how many people that you miss because they have too much mixed ancestry and that they don't fall into these groups.
Excellent question. So again, with the individuals who self-identify as Hispanic and Latino, they're included in that group without any sort of removal in terms of genetic similarity. For other populations, we usually, as part of general GWAS cleaning, and I'll say this is sort of the old standard protocol, was to remove individuals that are sort of one standard deviation from the centroid of that. And if you hold on for a little bit, I can talk about the centroids and the diversity here, but really we exclude not many individuals. So African Americans, we have a little bit more sort of increase in the inclusion criteria based on ancestry. So they do not have to be sort of predominantly African ancestry. Does that help answer your question?
Yeah, I think I'm just wondering, like, down the road in terms of implementation for like clinical care, like how many subjects will you be missing in the grander population because they don't fall into these groups?
Yep. That is a great segue to the real problem, right? Is what is the absolute risk for an admixed individual, right? And part of this is the genetic side of things, but that actually we can handle because we've standardized by all ancestry, the genetic similarity. We can take an individual from any ancestry, any admixture, and put them on a continuous scale like this and get a percent of their risk, right? Relative to their standardized sort of PRS distribution of what it would be for an individual with their ancestry. So we can do the genetic side of it. The complication is the baseline risk, right? So with using SEER data, it's the incidence and mortality rates are defined by race and ethnicity. So with what we just did here, everything is in sort of population bins. So what we really need are absolute risk on cohort data, right? We need individual level data where we can model the risk of an individual as a function of baseline. And we'll talk about what baseline really means here, and by the relative risk of that individual. We can peg that in terms of the population. And so we can do that with cohort data that's prospective, right? The most common model for that is a cause-specific hazards model. An example of that is a Cox model. It doesn't have to be the only one you use. And that's what we can do. But then we also need the data to do this, right? And this is where the multi-ethnic cohort comes in. So this is a cohort that was started here at USC and in Hawaii. In 1993, it includes about 215,000 individuals across 5 different race and ethnic groups. Over the years, Chris and others have done a series of nested case control studies to perform GWAS, to include them in a lot of these. And so we have about 34% of those individuals that now were included in the biobank and now have GWAS data on them, okay? And I get the case control numbers there in terms of the number of cases, in terms of colorectal, breast cancer, and prostate cancer. There's some complication in terms of the timeline, and we have to remove prevalent cases between this 1993 and 2001 period. But we can also construct a true incident cohort from the time of the biobank sampling onwards with this genetic data. Okay, so we can do this in terms of the true incident analysis on individual level data. And just to show where the individuals come from, this is Los Angeles, mostly individuals, the African-American individuals and Latinos from LA, and Hawaii has the Japanese-American, white, and Native Hawaiian individuals. Okay, now, every individual at the time that they entered the cohort was asked a question about their population, and the question here is, what is your ethnic or racial background? They were able to select one or more of these factors. Okay, and that usually put them into the 5 different main groups that we'll be talking about. There is another other category, which typically for MEC studies has been sort of ignored, but we want to include those going forward. We can estimate genetic similarity in these populations. Now, glossing over a little bit on the genetic similarity, but the types of variation that we can estimate is dependent upon the reference samples that we include. Okay, and so being sort of full disclosure, these are the reference samples that cover sort of superpopulations from African, European, Amerindian, East Asian ancestry, but also subpopulations within Africa from West and East, and other populations within East Asian. And from that, we can perform analysis for clustering. Now, with these plots, the x-axis here is the individuals that then we've ordered in terms of the self-identified race and ethnicity from the MAC. We estimate 5 different clusters in this, and then we can order them within those self-identified race and ethnicity groups, in terms of ranking of the majority of the cluster that we've assigned to that. Now, I am excluding, so typically if you had talked about this figure maybe 2 or 3 years ago, I would call these different clusters ancestries. Okay, and I would say this red cluster here, cluster 1, is predominantly in African Americans, therefore that represents African ancestry. And to some degree it does, but there is an uncertainty there about that. And it's easily maybe most seen with, if we look at the native Hawaiians, there's this orange cluster, cluster 5 here, okay? We don't have representation in our reference sample of Polynesian ancestry. We should suspect that this might represent that, but we don't really know. And there's an uncertainty there. And so I personally prefer to just leave this as a statistical approach, and we've estimated different clusters. But there's sort of ongoing discussions on when and where you can label sort of these clusters. But then we can also do the approach here through genetic similarity via principal components. And now I've identified, so everybody, the gray dots here represent the MAC individuals. The different sort of apex of the pyramids that you see in there represent the reference individuals from the different superpopulations from those from the reference data. And you can see then, and they're organized by the self-identified race and ethnicity groups within the multi-ethnic cohort. So you can see the diversity within individuals who self-identify as white in terms of the amount of diversity they have. And one to focus on here are the individuals who are equal distance between European ancestry and East Asian ancestry. So those are first generation biracial individuals, right? To focus on that genetic ancestry. But you can also look within the Japanese Americans and see a cluster of individuals that genetically are very similar, right? Those are also first generation biracial individuals, but now self-identify as Japanese Americans, okay, in terms of that. Now, it's also important to take in the social context of when the multi-ethnic cohort individuals, it's 1993, they were all individuals of 40 years plus introducing. So many of these individuals were born post-World War II, and that could influence how they self-identify as well, right? So genetically, they could be similar, self-identified, they might be very different in that. So something to consider as you think about, as we go forward here with the use of population descriptors. We can look within each population and look at higher level principal components to sort of abstract more variation. This is within the white individuals, and we can see that if we look on this scale, we have individuals that are from, there are a few MEC individuals that come from Hawaii, and those individuals tend to also have more variation to the East Asian populations. Yes. And then, oh, sorry, that's the difference. Those individuals are the LA individuals going to Amerindian ancestry, where the other axis here are individuals that go to sort of East Asian ancestry. So while these principal components are capturing substructure within the white individuals, they're also capturing geographic differences as well. And then we can look within the Native Hawaiians with different levels, and you can see that there's quite a bit of variation. I'll say though that visual display of the variation is really a function of all the levels of the principal components that we're seeing there. So just to take a little bit of time of sort of separating out these population descriptors and the genetic similarity in race and ethnicity, we have to be aware of sort of this new published report from the National Academies of Science, and it's really having a huge impact on the field. Just last week, we had a 2-day workshop about the use of population descriptors. And really, it's the use of population descriptors in genetics and genomics. And this is really following sort of the genetic field is pushing towards this continuum of ancestry. Instead of identifying bends, they're looking at this ancestry continuum, and rightly so, they're pushing that. And so the recommendations that come out of that are researchers should not use race as a proxy for human genetic variation. Researchers conducting human genetic studies should directly evaluate these environmental factors. And then researchers should tailor their use of population descriptors to the type and purpose of the studies. And they give several different types and purposes, gene discovery, and so on. The two that we're really focusing on today are trait prediction, complex trait prediction, and then health disparities research. And their guidelines in general say that similarity suffices for the genetic measure for trait prediction, but not all health disparity studies rely on descent-associated descriptors in population groupings. And that's really where I want to talk to about sort of the different modeling options that we can use. So if we go back into the MEC and we try to model these, model our relative risk, our absolute risk, we can do that with models like this. And we can fit a term, a single polygenic risk term, to the entire population in the MEC, because we can do that, right? And we get this sort of average here in the MEC at 1.92 for the 1 standard deviation increase. And then we also include in this model indicators for self-identified race and ethnicity. And we do that into relative to white individuals. And you can see that there's that about 1.8 increase in incidence for African Americans for prostate cancer relative to white individuals. So we can pick that, we see the same data and what we expect here in the multi-ethnic cohort for that. We can plot those in terms of absolute risk curves, cumulative absolute risk curves here. And so again, across the self-identified race and ethnicity groups, within each population, it's separated out by the genetic risk. So we have the individuals at the 20th percentile, 50th percentile, and 80th percentile. So you can see that, but each one of those, the differences within each group is the same, 1.92. It's the same relative 1 standard deviation increase there. So you can see individuals in African-American individuals that are at the 80th percentile in the multi-ethnic cohort have about a 35% chance of prostate cancer, okay, relative to the others. And what's driving this difference is baseline risk that's captured by race and ethnicity. So again, the question becomes, well, what about admixed individuals, right? So now we can get to this idea of including genetic similarity. So we can include that in a model rather than race and ethnicity as a variable. We can include genetic similarity, and we can construct curves like this. So it's a little bit busy here, but if we focus on the right, this is white individuals within the MEC. The blue line here represents what the absolute risk is when we use the race and ethnicity variable. The red here is when we use the cluster 4, which is mostly European ancestry, and we get very similar risk estimates there. Not much. There's not much variation within white individuals for that. If we go to the other side here with African Americans, we have the blue line representing what would happen with an absolute risk when we have self-identified race and ethnicity. And then the red line is individuals 100% for cluster 1 or African ancestry. And you can see there's slight differences there. And if we model sort of the average individual within the African Americans in terms of their admixture, which is about 85% cluster 1 and 15% cluster 4, we can identify a risk that's between those two extremes. Okay, so we now have the ability to model admixed individuals, or at least we think we do. Okay, if we look within the Latino individuals, Hispanic individuals, we see something slightly different. We see that if we model the entire population, it's the blue line, 100% of cluster 2, which is Amerindian ancestry, actually has risk that's between, okay, modeling the 50% one cluster or another. So it doesn't necessarily follow along the same sort of lines of the similarities and differences we saw within race and ethnicity. Okay, and that's because genetic similarity is not capturing the genetic admixture, it's capturing non-genetic factors. And so when we include it as a variable, we're capturing other things, right? And so this has come up in a lot of studies where people have been including genetic similarity and they model it with a risk term and they say this is capturing genetic risk, and it's not, it's capturing non-genetic risk. And so it's really related to all of the things that might be going on this other side of this DAG in terms of race and ethnicity factors that might be correlated with that and the other proxies that might be involved in that. And certainly we can take a look within the multi-ethnic cohort and we can see how these are all related. We can just pull out two variables that have been defined within the multi-ethnic cohort, years of schooling and neighborhood socioeconomic status. And we can see, well, what is the amount of variation that's described in those variables as a function of the PCs, the genetic similarity variable, or a function of self-identified race and ethnicity, or the combination of both of those. And so this yellow bars here represent when we take the entire MEC sample and we look at the amount of variation explained. And so we see that the PCs capture actually more of the variation of years schooling in the entire MEC than self-identified race and ethnicity. Putting both of them together, you don't explain much more. And we see the same sort of trend with neighborhood socioeconomic status. Okay. We can then stratify by race and ethnicity and look at the amount of variation that PCs or the genetic similarity explains within each one of those populations. And you can see that it's much smaller, but it does explain some of that variation. So about 5% of the variation within white of years schooling within white is explained by genetic similarity. So even within race and ethnicity, these variables capture some of these non-genetic factors. So what this suggests is that we should be modeling these non-genetic factors, right, in our joint models. And the distribution of those will capture the right risk for admixed individuals. So can we use the distribution of known risk factors to do that? And here we go back to a paper by Malcolm Pike. Those who've been around long enough know who Malcolm Pike is. Those who haven't, he's, for many of us, he's the reason we came to USC. So he was the former chair of our department, an epidemiologist, and world-renowned for his breast cancer research. And here he have used the MEC data to look at breast cancer and the difference across race and ethnicity at that time. And you can see that here with breast cancer, African Americans have less risk relative to white individuals, and Native Hawaiians have about 1.33 more risk, okay, in terms of breast cancer. Then Malcolm asked the question of, well, if we look at known risk factors at that time, this is back in 2004, and we model the distribution of those, so menopause, menarche, age of first birth, number of children, weight, HRT, alcohol, and we model that distribution, what does it look like in terms of the risk for all individuals? So this is sort of an earlier version of what we would now call causal modeling and GFG computation in many ways. You're taking the distribution of your risk factors, the estimates of relative risk that you've gotten from your data, and you use that to predict risk. And then you look at the risk difference between the populations. So he did that, and he ends up with these trends and seeing that the risk difference of just based on the distribution of these known risk factors explains, okay, those risk difference is the same from the observed to what's predicted in African Americans. So it explains that risk difference for African Americans. It actually increases, okay, or decreases the risk here for Native Hawaiians, okay? Which actually, if you adjust for these risk factors, the observed risk for Native Hawaiians increases in this case. So it doesn't actually capture that Native Hawaiian difference, okay? And Malcolm wrote about that, saying, well, another most interesting finding is the very high breast cancer risk that we observe in the Native Hawaiians, okay? The known risk factors are much less than the risk that's observed. And so he said that this could be a function of the number of high-risk alleles for these two genes. And those were the two genes of a few that were known at that time, right? in women from different racial ethnic groups. And he points out that 42% of the native Hawaiians have 3 or 4 of these high-risk alleles. Okay, where only 25% of the women in the other populations had those. So what he's suggesting is that we use the distribution of genetics to describe population differences, right? But the problem with our polygenic risk score is we haven't identified the causal variance. We've identified lots of LD and other things, so we can't interpret this distribution. And we would like to, but we can't. And in prostate cancer, there's very few non-genetic risk factors that we know about that we can use even in these joint models. So if we keep this distribution and we try to model it in our sort of way that we've done before, we put this distribution into our multi-ethnic cohort analysis with all individuals, and we just use their raw genetic score, this is the figure we get, right? It does not explain the incidence differences we see. Okay, it does a little bit. There's a slight increase here in terms of the African-American risk, but it's not good enough, right? If we use this for predicting risk, then that would lead to health disparities. So what it suggests is at this time, the best thing to use for health disparities research in the MAC for prostate cancer is to use race and ethnicity as the baseline risk, right? And to go forward with using other population descriptors and other risk factors to sort of, and this is where it gets to the point of sort of the environmental components, because can we add in exposome research, metabolomics, other biological factors that we can also model within the multi-ethnic cohort to better capture this risk. So in conclusion here, hopefully I'm pointing out the importance of a diversity within a single prospective cohort. So a lot of the work that's been done comparing cohorts and incidence rates to date, even in genetics, most cohorts are still within self-identified race and ethnicity bends. We think of the Southern Community cohort, which is mostly African-American. In other cohorts, there's Asian cohorts and others. When you compare those, they have differences in terms of geography, risk factor distributions, and other things. And so we really need more cohorts like the MEC that are diverse in a single cohort selection and location and geography and how to look forward to that. Now, there are biobanks that are pushing towards this, but most biobanks are selected off of health systems. And so are they true incidence cohorts? And there's a lot of work being done of how you can do subsampling of these biobanks to really represent incident cohorts. There's also All of Us, which is supposedly a population-based sample, but supposedly is the key word there because there's a lot of selection bias that goes in there. The other things to think about is when and how a PRS distribution can be used. If we truly know that we have the causal risk factor, the causal variance, then maybe we can use those distributions to describe some differences. But if it depends on sort of if we're using all of the genome-wide data to predict risk, then I think we can't. Most of the techniques right now for predicting polygenic risk scores are going towards using genome-wide data, machine learning techniques, high dimensional data analysis, deep learning, to really get a polygenic risk score that's used to capture the entire genome. Okay, there are pros and cons to that. One being also is how would you pull out PSA snips from that or something along those lines and that type of thing and use that. So there's also careful use and interpretation of the population descriptors. We need to be very clear about why we're using these population descriptors and what they're capturing. Okay, when we use genetic similarity as a population descriptor, what is it really describing and why? Okay, and then again, how do we model risk for admixed individuals if we can't have a fully described joint model that has all the known risk factors or something and it describes those risk differences? And then finally, for this group, the incorporation of environmental factors or social determinants and proxies. How do we incorporate those just as variables in their distribution? There's a lot of work now about including them with interactions. But again, the same thing needs to be thought about. So from the genetic side, a lot of people are doing work on sort of looking at polygenic risk score by genetic ancestry, genetic similarity interactions for heterogeneity across ancestries. This gets at the idea that the construction of the polygenic risk score is not portable across ancestries. We don't have the right SNPs, we don't have the right LD, whatever it is, we can't use that. But remember, genetic similarity is related to race and ethnicity. If we do a polygenic risk score by race and ethnicity, what is that capturing? If we do a polygenic risk score by social determinants of health, there are now lots of papers coming out for that. What does that actually capture if we do that? Is it population differences? Is it genetic differences? Is it also gets into subtle issues of heritability and genetic heritability as a term of the amount of variation that's explained by genetics. This has been known for years that you can't actually compare those estimates across populations. But what does it mean? If it can't compare them, then you'll see heterogeneity because it's just based on the amount of variation of that trait in a population. So, and also just, I want to highlight the NASM recommendations here in terms of the different types, and I don't have much time here, but just to focus on, for this group, trait prediction. And if you follow this through in terms of their recommendations, and they give slides here, and the use of genetic similarity always comes up, they're pushing this, right? And then at the bottom of it says, well, if you want to incorporate environment, it's down here, whether gene environment interaction or what, see their environment slide. Okay, and you go to the health disparities and you go through this paradigm and you choose this in terms of understanding health disparities mediated by social processes or the study of the impact of environmental exposures. It all leads to the environment. And if you go to that slide, it basically says measure them if you can. Okay, and if you can't, then carefully consider the use of proxy variables. Okay, and so this is where I think environmental exposure assessment is the measuring if you can. We need to get better measures of social determinants of health. We need to get better measures of the burden of environmental exposures so they can be used in these joint models so we can better predict risk across populations. So that's the end. Thank everybody who has contributed to this, and this is really leaving people out. But certainly Chris for all that he's done with the prostate cancer, the data. David Bogomil is driving a lot of the absolute risk within the MAC, and then Sylvia and Anshi Sylvia's been working on the methods for discovery and PRS construction, and Anshi led the latest GWAS. So with that, I'll stop and see if there's any questions. We'll let, you've already asked, Rob.
But no, thank you for this presentation. So I guess in a certain way, right? Like I'm talking about the race and ethnicity sort of descriptors. I'm wondering whether for MEC, it seems like there was a lot of effort kind of put into choose specific labels for Japanese American and Native Hawaiian. Is Hispanic the right level for that kind of variable? Especially if they were kind of collected mostly in LA. Like, is there are there more Mexican? Are there more Central American? And how
yeah, so
break that down further?
Yeah, they did ask sort of acculturation questions and migration questions of what generation they are. Plus, we can also do more fine scale ancestry type estimates for that. So most of them are of Mexican ancestry and have first or second generation from Mexico. So it's a little bit different, but that's again, the broad population classification that's used.
Yeah. Well, in another sort of, and I think that needs to report also, but also on the other side, from how do we use these races and ethnicity variables, right? There also is always a strong recommendation on, especially for reproducibility and communication, right? That we shouldn't take for granted that Hispanic is the label that's appropriate, right? And the specificity is important. And so I'm just wondering, and so I guess it was would Mexican American with some variation, right, actually be a better descriptor than Hispanic?
Probably a better descriptor. But
part of it too, so this is a lot of what we were talking about the other week at what was it?
The workshop,
Workshop. Is the multi-ethnic cohort falls into what people call legacy data. So, I mean, at the time, you saw the survey he showed, people saw the options. And I mean, it's a combination of two things. It's a combination of what do people feel like they should select from the survey options, and then also what options were they given when the survey was created in the early 90s.
Yeah, and there's a big issue of legacy data versus how we would do the study now. right, going forward, and can we recontact, can we ask these questions differently, can we, you know, do that?
Most meta studies, when they analyze the data and present the results stratified, they present it by race, ethnicity. But race and ethnicity are two separate terms, right? So there's overlap between them, but here they're kind of thinned. And on top of that, there was like a binning algorithm where somebody answered white and African American. They're classified as African American.
Yeah, and I think the question that you were showing, that was the one that was presented to them, that definitely makes sense.
Yeah.
It's an ethno-racial construct, I mean, she's
Yeah.
sort of within this, but in the categories that were presented, there is like, there's more specificity and
Yeah.
like it did say Japanese or Native Hawaiian, but it didn't indicate whether it was Japanese American or it was just Japanese sort of ethnic heritage. Right? And so like, there was a decision sort of made to describe it with more specific, not Japanese, but Japanese American, but that isn't, that same logic isn't applied to the Hispanic.
Yeah.
You know what I mean?
Yep.
Like
And there's more work within the Japanese Americans, you know, identifying Okinawan versus not in by
Yeah.
name, and so, you know, but it's something that could be and should be pursued.
And then also too, this is something that we discussed now, whether you go into legacy data, if somebody self-reported African American, is it correct to recode them based on other things, right? So, and put them in a new group based on how you think they should be. They said they were African American, but is it right to categorize them? So I mean, with Latino, right, so that's ethnicity, would it be correct to try to racially categorize them?
Yeah, so, yeah, and I get just very specific for like the Japanese American, because not taking into account, like Japanese American population. That taken into account generation of like immigration. So in that case, that label is different.
Mhm.
So I guess I'm just saying that like. Even though that it is legacy data, we do still make decisions about
Yeah.
the recoding and everything.
Yeah.
Rob, did you still? Rob and then
Yeah,
Erica?
mine's a little related to this.
Oh, okay.
So, so it sounds like the conversation was about, that we were just having was around, you know, how you label things, how you survey, how it was described in the questionnaire, that's what you have some legacy data. How do you think about this? Because, you know, you have race, ethnicity, and you have these other factors that we know are correlated with that, they're correlated with the genetic factor, but they also are a snapshot of the time and place of people who had a certain lived experience. If you do that same study, you know, that was in the 90s, which apparently is about 30 years ago. But if you do it today, even if you have the same individual who might identify in the same category, let's say you use the same ones, that person's lived experience may be very different than what you're seeing in these data. So how do you think about that in these kinds of analyses, right? Where you're trying to look at these other factors, how you try to pull them out, you try to disentangle them, but time goes on, people's experience goes on, society changes, and now let's say 10 years trying to use some of these analyses to predict risks, but in a population that is very different than the population from which they drew from, which is that social context.
Yeah, the generalizability of the cohort, the age and also age time cohort issues are all at play here, right? And you're touching on all of those because the risk you know, when we look at the risk distribution and we describe it at that time point for those individuals who you had a very different, you know, lived experience, and shortly after, well, actually during, right? HRT as an example for the breast cancer, right? Recommendations have changed over time, right? So the distribution and the use of HRT has changed, and, you know, so that's going on as well. And so, yes, it's a huge issue of how we use legacy data, older data to create risk models that then would work, right? And so I think you know, the hope is to develop these models and then, you know, can we go into all of us? Can we go into some of these newer cohorts and see how well they perform by age? You know, and it's not just the non-genetic factors that shift. PRS, the distributions then the allele frequencies haven't shifted in just a short period of time, but we do see that polygenic risk scores consistently have bigger effects for younger age of onset. And so the application of the PRS, and I just showed sort of that one standard deviation, you know, some of the other work we're doing, it actually matters about the application of it. You would do it with an age interaction, but also the genetic distance that they might be in terms of to the data that was used for discovery, right? If you use mostly European, then you could see that they do, if they have a genetic distance from that, they might perform less well for that person, but, or population, right? And then that's how do we capture that? So there's a lot there in applying these. You know, so there's the Emerge Consortium is now starting to do this and use this as sort of how do we apply these models for population level screening, and or develop models that would be used for treatment decisions as well. And all of these are the issues of complication, right? Yes.
Just tying into this, you mentioned genetic disease, but you're also using data of self-report. So how are you working with those together? They're very different, right?
Well, I mean, so they are genetic, I mean, the genetic ancestry, right, is highly correlated with that, with race and ethnicity, because,
How do you get
sorry,
their genetic?
how do we, so this,
How do you get their genetic history?
well, so.
I mean, taking, because your first slide was saying, of African descent or European descent, how is that obtained for each segment?
Well, that's, okay, so that's where we, when we look at sort of slides like this, right? And we look within sort of the individuals, so within African Americans, and then we have reference populations that are sampled from Africa, right? And so now individuals that are sampled from Africa, and you can see that the MEC individuals lie along this sort of cline here, representing sort of individual admixture between African and European ancestry in that realm. So then those individuals self-identify as a group based on our questionnaire in the MAC, but then they have genetic and admixture and then descent through that, okay? Through that similarity is what we're kind of inferring, and that's that admixture there, right? Is that helping?
Yeah,
answer your
I'm just
question?
trying to, I think so. I don't know what's ready. That's it. One question I just have is,
Mhm.
you were thinking about going forward. I guess, it's just the issue to self-reporting is so complex.
Yep.
And we have same families where a dad and a mom will probably say different things about their child.
Yep.
Right?
Yep.
For going forward, does technology exist to, omit that, to find that genetic history that, you don't have it now, but it exists, right?
So, exactly, so technology exists to put people in this sort of genetic similarity realm and call that, we label it ancestry in some realms if we have the right reference data. And so we can do that, right? That's what 23andMe does, right? The self-identified label part though, should it or not, and how does that then change over time? Most of the individuals in the MEC, even though they were given the option to select one or more bubbles, they selected one. Even if they were genetically admixed, they selected one value, right? If we perform that, survey today, that would probably be much different for people selecting multiple, it's much more common now to select multiple bubbles. And that might overlap with their genetic ancestry similarity that we estimate, and it might not, right? And so that's part of the nuance of what we, the take home message is, you can use genetic similarity, you can use self-identified race and ethnicity, but you need to know what you're capturing with those, right? That's and really, often we gloss over it and we just, oh, we got it, we use it. But we really need to be deliberate about what it means, what it's capturing, and understanding more about that downstream, especially when doing risk prediction. Any other questions online? In the room?
So,
Rob?
we're talking.
Yeah.
I'll just, I should have added a slide to the end. The Children's Center has this series, Be Heard, the tools for having more impact from your papers, and we have one more session next week, Friday at 10. And John Samet, who used to be the department chair, and Lisa Barros here will be talking about what to do when your results make industry mad. So, a lot of the people in this room has had experience of publishing a paper and then being the subject of attack from various industries. And John maybe has the most experience of anybody that I know in dealing with that. So that'll be the top. And I know some people here in this room have had recent experience with this. The new strategy, at least for some local industry, seems to be to target junior investigators. I don't get much from them anymore, but occasionally somebody comes into my office really flipped out because they got some really nasty letter from a lawyer or from another scientist. So I think I think it'll be a good session. Everybody should come. I hope you all does everybody get flyers for that? Does Domenica spam your inbox for it? Yeah, good. All right. Do we have lunch?